if you look at the other example of active transport, right here, so this is also a process that requires energy. You're going against a concentration gradient. So this process requires energy in the form of ATP. So when the molecules are brought in, ATP is broken down to ADP, and that energy is used to bring these molecules in. So these purple circles that come in, again, they're coming in against a concentration gradient, and this requires energy. The third example of energy requiring transport mechanism is group translocation. Now, in group translocation, the molecules are brought into the cell, and as they come into the cell, they undergo chemical modification, right? So they are chemically modified. So these blue triangles that enter the cell, they would be modified uh, commonly by the addition of a phosphate group, right? So you see this P that is attached to the blue triangle. So the P has been modified, sorry, the blue triangle has been modified by the addition of a phosphate group to it. Now, it is believed that bacterial cells, they carry out group translocation. They the most common molecule that they perform group translocation with is glucose. All right. So you can imagine the blue triangles are actually glucose. So what happens is glucose is taken into the cell and it is chemically modified. Now, it is believed that the bacterial cells do this so that once the molecule has been chemically modified, it will not leave the cell. It's sort of trapped inside the cell. So this is the cell's method of basically saving for a rainy day. So while there are glucose molecules on the outside, so you take them in as much as you can, and then you quickly modify them. You change them chemically such that now these molecules are trapped inside the cell and they can no longer leave the cell, which is a clever mechanism. Now, so we looked at how cells take things in, right? Either by facilitated diffusion or active transport or group translocation. Now we're going to look at how cells send things out, right? Which is called secretion, right? When cells produce something and they send it out of the cell, it is called secretion. Now, cells produce, especially bacterial cells, they produce a huge variety of proteins for a huge variety of reasons. Now, but a lot of these times, the proteins are relatively large, right? So as you can see, this is a very large enzyme that the cell has produced to be secreted. But an enzyme as large as this, there is no way it's going to able to simply diffuse through the plasma membrane. Likewise, it is definitely not going to fit through a protein channel like this. So what happens is when the cell produces a protein that it wants to secrete, that it wants to send out of the cell for whatever reason. So, for example, in this case, the green enzyme that it has produced to send out of the cell is an enzyme that performs some sort of a hydrolysis reaction where it breaks down a large molecule into tiny molecules which can then be taken into the cell, right? So you have food that is outside the cell, but the food is too big to come in. So you produce an enzyme, you send it out of the cell to break the food down into smaller pieces, and these smaller pieces can come into the cell. Unfortunately, the trick here is the enzyme that is supposed to do the job itself is relatively large and it can't simply diffuse across the plasma membrane, and it cannot also simply pass through the protein channel. So what the cell does then is, remember enzymes, they have very specific shapes, right? They have a specific shape for the specific function that they do. Now, the cell produces the protein molecule in a linear, all right, in a linear format. Okay, and one end of the protein molecule, the protein sequence, you have a signal sequence. Now, this signal sequence, which is that orange bit, is the part that tells the cell or the protein channel that this protein, this pre-protein, has has been made and it is destined to be sent out of the cell. So this protein must not be kept inside the cell. The function of this protein is outside the cell. So please send this protein out of the cell. So that is the job of the signal sequence. As you know, proteins are made up of amino acids, right? We have 20 different types of amino acids. So these amino acids, they are all synthesized in chains. Okay, so they're all first synthesized in chains, and these chains can be hundreds and thousands of amino acids long. Now, once they are synthesized in such a long chain, then they are folded to give the complex protein shape. All right. So first they are synthesized in a long chain, and then they are folded later. For proteins that are supposed to be sent out of the cell, the end of the protein sequence, you have an additional sequence called the signal sequence. So this signal sequence would be the indicator to say that this protein must be sent out of the cell before it can be folded. So it shouldn't fold inside the cell, because if it folds inside the cell, then the structure becomes too big for it to exit the cell. So the signal molecule makes sure that this pre-protein is sent out of the cell before it assumes its specific three-dimensional structure. So that's what happens here for this green molecule. And this orange bit is the signal sequence. And before the protein folds into its specific shape, it's called a pre-protein. Um, right? It's called a pre-protein. So the pre-protein is sent out through the protein channel. That is possible because you're sending out just a chain of amino acids, and this chain of amino acids can very easily squeeze through this protein channel. So it is sent out. Now, once it is sent out, that's when it folds into its specific shape. It performs its job, and in this case, it's a hydrolysis function. It breaks this very large food molecule down into smaller pieces, and these smaller pieces are then taken into the cell for metabolism.